Welcome back. Last time we discussed the different formed elements that we find in the blood. Today we'll get to look at what drives some of the synthesis of those particular formed elements. The whole process, no matter which formed element we're talking about, falls under the grand heading of hematopoiesis. In the big scheme of things, we see all formed elements from the blood, whether they be red blood cells or our various types of white blood cells or our platelets, all come from a primitive stem cell, if you will, called a hemocytoblast. Regardless of what the blood cell type is, that's our origination point. Now, the process through which we go through to the endpoints are going to vary wildly. We'll talk about some different tissues that we see different types of blood cells form in, and certainly what drives their synthesis is going to vary wildly, simply because the function of each of those types of formed elements is going to vary so much. When we look at red blood cell production, we're going to call it erythropoiesis. Now, going back to what we discussed last time about red blood cell function, its primary function is going to be to carry oxygen, carbon dioxide, to and from the tissues. That's going to occur primarily in the myeloid tissue of the red bone marrow, most notably sternum, ribs, femur, humerus, vertebra, and pelvis. This is going to be driven in a negative feedback type of manner. Now, remember when we talked about negative feedback back in A&P1? Negative feedback always looks to take a deviation away from an average set point and bring it back to the normal. So we will see here on the left, we have temperatures, because temperature in the room acts as a negative feedback. But if we start to see red blood cell production get a little too elevated, oxygenation a little too robust in the blood, we will see a decrease in red blood cell production. And then we will start to see blood levels drop. They will drop to a certain point. Then we will see red blood cell production start to pick up again. But this is going to be driven based on what the receptor level of our feedback system is telling us about the oxygen carrying capacity of that blood. Whenever we want to increase red blood cell production, we turn to a hormone called erythropoietin. Now, in everyday publications, you may have come across it simply abbreviated as EPO, because it is one of those hormones that pop up in everyday articles from time to time, especially in the world of athletics. In cycling in particular, any endurance sport, but cycling in particular seems to be most notable for it. The injection of synthetic erythropoietin tends to be one of the more known ways of getting around natural abilities. As you can imagine, red blood cells being noted for their oxygen carrying capacity, and anybody who's ever run the length of their driveway knows that you need oxygen capacity to carry out endurance activity. So if we could get the red blood cell count elevated, increase the oxygen carrying capacity of the blood, you should be able to perform better in endurance sports. And that is simply what cycling has seen over and over and over again. Now, you can induce this naturally. If we take you from the elevation that we typically experience in Kentucky and bring you to Denver for a ski trip, the first thing you're probably going to notice as soon as you get to whatever cabin you're staying in is you're going to be winded simply taking the bags out of the truck and bringing them inside, because the air is so much thinner in oxygen composition out there. So in order to get the same amount of oxygen, you need to increase your respiration rate. Now, this is going to be taken care of by that negative feedback system. Your body's going to recognize this and step up red blood cell production. It's going to increase erythropoietin secretion, drive that red blood cell production from the myeloid tissue of the bone marrow. Increase red blood cell production up to a point where the carrying capacity now compensates for the low density air that you happen to be breathing in. Now your respiration rate drops back to normal, and you can get the oxygen you need out of the density of the air that happens to be available. And you see endurance sports athletes take advantage of this as well. They may train at high altitudes. You train for the Boston Marathon, which is going to be very close to sea level. You train at high altitudes. A couple of days before the race, you come down to sea level, and you should have that increased ability. Now, people who do that, I mean, the average Joe, you're not going to notice much of a difference doing that. But if we're talking about a high-end athlete, and now we introduce the opportunity to synthetically inject EPO, and you can take that to the next level altogether. Now, of course, the production of red blood cells also relies on a lot of other raw products, iron in particular. We mentioned hemoglobin last time, that 4-chain protein with that iron-centered heme molecule as part of it, in which the oxygen molecules are going to bind to. If you are deficient in iron, if you're deficient in certain amino acids, folic acid, B12, you're going to lack the raw materials in order to synthesize the red blood cells. So we see this ebb and flow, that sinusoidal wave that we typically see with any negative feedback loop occur with red blood cell production. As oxygenation drops down, we see erythropoietin from the kidneys being secreted. The dry myeloid tissue to produce more red blood cells. Oxygen carrying capacity increases. Once we get an imbalance in the opposite direction, erythropoietin secretion shuts down, and red blood cell production drops down with it. We typically see red blood cells last in the blood about 120 days or so. But we see a lot of recycling of these raw materials. When a red blood cell breaks down, we take the hemoglobin and we break it apart into heme and globin, as if the name wasn't already suggestive of that. The heme is that more inorganic, iron-containing molecule, with the globin is going to be more the amino acid protein structure. We take that globin and we break it down into its amino acid subunits, and we can dump those right back into the blood again. No different than if you had ingested a protein and absorbed the amino acids as part of its subunits. The heme, on the other hand, now goes into more of a breakdown. We remove the iron, leaving behind bilirubin. The iron is going to be bound to a protein called transferrin, and that's going to be released back into the bloodstream, just like it happened with the amino acids from the globulins. Now, this is all processed in the liver. The liver now will take the bilirubin, the heme portion without the iron, and it's going to dump it into the intestines. And it's actually that bilirubin, as long as the liver can get it out of the blood and get it into the intestines, is what gives species its more brown color characteristic. A decrease in oxygen carrying capacity secondary to red blood cell production or abilities is going to be considered an anemia, and there's several different types. First off, we can have a nutritional anemia. A couple of slides back, we had a couple of things that you would typically get from the diet. Folic acid, for example, iron. If you're simply not getting raw materials from the diet, you're going to have a pretty standard nutritional anemia. So your red blood cell count is going to be suppressed simply because the diet is inadequate, malnourished. Sometimes considered a subdivision of those nutritional anemias would be pernicious anemia. Now, this is going to be specific for a vitamin B12 deficiency, which, of course, you can get from your diet, hence it's linked to nutritional anemias. The B vitamins, however, can be notorious for being difficult to absorb across the GI tract. Many of you listening might have already had various vitamin deficiencies and you were given an oral vitamin to take. Most vitamins are very readily absorbed across that GI tract. For some reason, B vitamins seems to be more of an obstacle. So a lot of people who may be deficient in B and its various subtypes, typically it's delivered in an injection, bypass the gut altogether. But B12 deficiency in particular would be more of a pernicious anemia. But recognize that if it's a gut problem, you may be ingesting adequate amounts. You're just not absorbing it. Next up would be sickle cell anemia. Now we're getting into more of a genetic disorder. This is a recessive trait that you can have the gene for it, but not express the physical features. And when we say sickle cell, really what we're referring to is a deformed red blood cell, where it simply takes on a curved sickle shape. If we have two parents who happen to be a carrier, remember it's recessive. In order for you to express the sickle cell characteristics, you have to have those genes from both parents. So if we look at the offspring of those parents, you will have, on average, one person who is completely unaffected, 1 in 4. He gets the normal gene from both parents. You will get 2 out of 4, or half, they'll be unaffected carriers. So you have one who's completely unaffected, doesn't express the sickle cell physical characteristics, doesn't carry the gene into further lineage. Half, however, will be unaffected. Their cells will still be normal, but they carry the gene. So they have the potential to still pass their recessive gene onto any offspring. And that's only going to be of a consequence if they partner up with somebody else who also has the recessive gene. Because if we partner up with somebody who, brother, sister, we partner up with somebody else who is a non-gene carrier, you will never develop the recessive traits because you have to have the recessive gene from both parents. This would be the person who would be affected. Not only now are they gene carriers, but because they possess both recessive genes, they will exhibit the sickle cell trait. Now, looking at the shape of that, remember back to a couple of videos that we showed you last class. We had red blood cells flowing through capillaries, and we made particular note of the importance of a flexible membrane. We talked about sharp bends in capillary beds and the collisions that these red blood cells take on as they go through these vascular trees. A flexible red blood cell membrane reduces the chance of it being damaged. It allows it to squeak through tight spaces. It allows it to squeeze through tight capillaries to keep that tight contact with the capillary wall to increase the ability to diffuse gases in both directions. The sickle cell appearance is going to cause any number of problems just for that reason alone. Not only do these cells carry less oxygen, their carrying capacity is less, but they exhibit all sorts of problems simply flowing through the vascular tree. And here on this diagram, you can see how we're getting a blood clot form. Right, here was the video we showed you last time. Now picture a significant percentage of those are sickle cell in shape and simply not able to round some of the bends, getting tangled up with each other and blocking up the vascular tree. The loss of that flexible membrane means now not only do these red blood cells carry less oxygen, but even the oxygen that they have, the rate and ability for it to diffuse across the capillary wall is going to be diminished because you won't get this nice, tight, seamless fit. So, red blood cell production, hemopoiesis, erythropoiesis, driven by the oxygen levels in the blood. Oxygen levels drop 2 down, erythropoietin is secreted to drive red blood cell production up, and vice versa. Leukopoiesis, being more associated with immune responses, are obviously going to have a different driving force. And that's what I really want you guys to focus on when it comes to this material, is those feedback mechanisms. Leukopoiesis, white blood cell production. Of course, we're talking about the full spectrum of white blood cells here. They're still going to originate from a common precursor cell, that hematocyte. We will tend to see the granulocytes, remember the neutrophils, the eosinophils, the basophils, and the monocytes are going to be produced in the myeloid tissue. The myeloid tissue is going to be found in the bone marrow. However, the lymphocytes, they're going to originate in the bone marrow, but then they're going to form colonies in lymphoid tissue where they continue their maturation. And when we talk about lymphoid tissue, we're talking about lymph nodes, tonsils, spleen, thymus, and well as some of it happens to be residing in the red bone marrow itself. So it might be hard to read this on the screen because of the text and the color, but again, have a look at the notes and see the difference between myeloid cells, myeloid tissue, versus the lymphoid cells and lymphoid tissue. We will definitely get into these chemical messengers that drive leukopoiesis more so when we get to immunity. But I want you to just get a general picture of what's going to happen here. If you get damage, tissue damage, if you get the inflammatory changes that are going to take place with various infections, the tissues local to that site are going to send out chemical messengers. Cytokines, which some of those are various interferons, lymphokines. That is going to trigger a migration of specific white blood cells to the area where they're needed. Because unlike red blood cell production, where we're going to need that elevated red blood cell production all over the body, we want white blood cells to be a little bit more targeted. We want to go to the site of infection. So they're going to use a process called positive chemotaxis. You have the injured site, you have the infected site secreting these chemicals, resulting in increased white blood cell production, but also targeting their migration. Think about a shark. You hear ridiculously special character traits about sharks on how they can detect blood in the water, like 1 part per billion from a mile away. Just outrageous numbers. A shark is able to hone in on the source of that because he's continually moving in the direction of higher concentration. He can detect that 1 part per billion, but now he heads off in that direction, a quarter of a mile, closer to the source. Now the concentration is going to be up. So he knows he's moving in the right direction as long as the concentration of that source chemical keeps getting higher. And that's similar to what we see with that driving not only white blood cell production, but white blood cell migration. So still originating from the hemocytoblast, in the myeloid tissue for the granulocytes and the monocytes, lymphoid tissue for the lymphocytes, to give us our array of different white blood cells with their physical characteristics and their particular specialties. So for now, again, we don't want to get into immune function too deep now because it's coming up in its own separate topic. Focus on red blood cell production and the negative feedback mechanism within. Focus on the anemia types. And again, to reinforce the importance of the flexibility of that white blood cell.